So we'll move to our next session. It's a technical talk session. So this is 20 minutes per speaker, and please control your time. And I will probably remind you if you're close to your time, and including Q&A. So our first speaker is Dr. Slominski from Department of Dermatology at UAB. He will talk about the recent advances in vitamin D signaling, new opportunities for computational biology. So probably you can share your screen now.
Yeah, I have shared the screen. Do you see the screen?
Not yet. So you need to click the button. There's a bottom. There's green share screen button. And
yeah,
then...
I have done this. Okay, let's see. Do you see it?
Yes, yeah, I can see it.
Now you see it, and I have to put probably for presentation. Okay. Let's see. Do you see?
Yes, I can see it.
Okay, so I have to move this. Okay, or maybe to put on the bottom. So I will be speaking about the vitamin D, and the vitamin D is produced from the 7-dehydrocholesterol through the simple photochemical transformation, and the transformation after the absorption of the UVB by B ring of 7-dehydrocholesterol configuration pre-vitamin D3, which in the room temperature change into vitamin D3, more UVB energy. There is a further isomerization with production of lomisterol and production of the tachysterol. So these are different compounds. I'm not sure how to move this. How to move this?
Just click the arrow is fine. The down arrow, or click the.
Okay. Okay.
Yeah.
So then this is the slide which really I've obtained from Michael Holick, who is a guru in this vitamin D field. So again, production, this is the skin, UVB, production of vitamin D, which is entering the circulation. Vitamin D is also delivered through the food, through the alimentary tract, transport to the circulation, and then is going to the liver. In the liver is hydroxylated in position 25, again entering circulation. This is a classical dogma. Then entering the kidney, in kidney C1 alpha hydroxylation. So we have 125 dihydroxy vitamin D3, which by dogma was considered as the only one form of the active form of vitamin D3, enter again the circulation, going to the target organs and interact with the vitamin D receptors. So dogma another was only one of vitamin D receptors. And we broken this dogma by discovery of novel pathways of vitamin D activating pathways. So we have revised the traditional we have revised traditional role of CYP11A1, the rate-limiting enzyme of steroidogenesis, which is expressed in addition to adrenals, gonads, and placenta, is also in the skin, and it metabolizes 7-dehydrocholesterol, vitamin D3 and D2, and lumisterol, with intermediates being biologically active or circulating in the serum. So we performed several experiments, and we detected many of different compounds, many of these compounds in the human serum and human epidermis. So several biochemical and chemical analysis allowed us to really lay down the following pathways. So CYP11A1 is acting on vitamin D, is hydroxylating in different positions. Different positions are by numbers, so there are different carbons on which it is hydroxylated, and we know OH or 2OH, so 2 carbon, 2 hydroxyl, 20, 22, for example, position. But the main product is 20-hydroxy, which is further also hydroxylated by CYP11A1 to other hydroxy derivatives or by other enzymes to produce at least 15 new vitamin D hydroxy derivatives, which have a different position of hydroxy group on the side chain or D ring. This is in the skin is sometimes even more complex because 7-dehydrocholesterol, which is precursor, is also substrate for CYP11A1, producing 22 or 20 hydroxy or dihydroxy, or be cleaved side chain to produce 7-dehydropregnenolone. And these compounds, under the UVB, they change configuration into the vitamin D or lumisterol compounds, or vitamin D-like compounds with short side chain, giving even much larger number of the compounds that can be produced in the body, whole new metabolome from lumisterol and vitamin D and tachysterol also. So, in conclusion, dogma that vitamin D is solely activated through 25 and 1-alpha hydroxylation is invalidated by our discovery of novel pathways of vitamin D metabolism by CYP11A1 with multiple hydroxy form, and the same is for the vitamin D2. Okay, these pathways operate in vivo. They are initiated by action CYP11A1, and main products including 20-hydroxy vitamin D are present in human serum and epidermis. The detection of novel vitamin D3 metabolites in the skin, adrenal gland, and serum in conjunction with their biological activity indicates that they act as hormone in vivo, for which the effect would depend on their local and systemic production and their metabolism. And here are on the examples, because we publish a lot. So induction of cell-differentiated flow cytometry, immunocytochemistry, involucre. Then action on some regulatory pathways, NF-kappa beta pathway, important in inflammatory responses. So 20-hydroxy vitamin D3 is downregulating NF-kappa activity, and this is the mechanism which we have proposed previously. So we are at the stage of preclinical testing of anti-cancer activity of different calcemic analogs. So what was done in vitro test, proliferation, colony formation, soft agar, any other anti-tumor test. We already know about the toxicity. We compared 125 with the 20-hydroxy, and 125 is toxic is because of the hypercalcemic effect. This is a kidney, this is the heart deposition of the calcium, small amounts when treated animals, and no effect in the heart and kidney of this compound. And then stage of the in vivo testing. So this is like a 7-8 months experiment. It's a UVB-induced skin cancer. So skin cancer induced by UVB and treatment by subcutaneous injection of 10 micrograms of 20-hydroxy vitamin D 3 times a week have reduced the tumorogenesis, reduce the number of tumors, reduce the total volume of tumors. The animals were killed, and then we analyze, I have analyzed, I am pathologist, the different organs, no sign of toxicity. So injection of human melanoma into the nude mice and treatment of 20-hydroxy vitamin D at dose 30 micrograms intraperitoneally, so the inhibition, there is inhibition of tumor growth, subcutaneous tumor growth. So then another examples, we really started to look on the photoprotective effect, and this is the DNA damage measurement with the COMET assay, and which shows that all of these compounds protect against the DNA damage. And this mechanism includes the activation of NRF2 and P53 phosphorylation. This was published in a few other papers, Redox Biology, but this also the concepts of vitamin D hydroxy and lumisterol through the action on NRF2 and downstream enzymes inhibit the reactive oxygen species, then the DNA repair mechanism through the P53 phosphorylation and translocation, DNA repair inhibition of carcinogenesis. But let's go to mechanism of action of novel corticosteroids, where it is ligand receptor interaction. We look at the nuclear receptor. Classical receptor is a vitamin D receptor, and this is a crystal structure. It was done by Whaley and my collaborator Whaley and Natasha Rochelle from France, and I'm the author but it was led by Whaley. They co-crystallized with the ligand binding domain. The control 125, classical. This is another compound which is produced by CYP11A1, 120 hydroxy, which has a little bit similar structure as 125 and 20 hydroxy. So as we can see, I will not go in detail because you can find in the papers, but the interaction in the ligand binding domain 125 and 120 is very similar, but this interaction are very different. With some similarity for 20 hydroxy vitamin D3, which may have the important implication for signaling. Then we look on retinic orphan oxide receptor alpha and gamma modeling. It was done originally by Whaley, and the new vitamin D compounds, they really bind to the ligand binding domain in similar manner as the native ligands. And the compounds were sent to NIH, to NIHS, to Anton Jetten. I will not go in detail on this. I will just, it is also published. So he used his models and checked the transcriptional activity downstream of the ROR alpha and ROM gamma. Conclusion is, and it's unquestionable, the novel hydroxy derivatives of vitamin D3 generated by CYP11A1 act as reverse agonists on the ROR alpha and ROR gamma. Reverse agonist means in the inhibition of transcription of genes downstream of this receptor after activation of the receptor. Then we performed, so first was performed microarray analysis, and which was David Grossman did very good analysis ingenuity pathways, and this is for 2023 new compound versus control, the classical. So there are huge differences in the transcriptosome. But what was very interesting coming out, ingenuity analysis for classical 125 dihydroxyvitamin D3, number one receptor is vitamin D receptor activating. And this is another after 24 hours because we did the time dependent. Vitamin D receptor. But what was coming, interestingly, second receptor on the line was aryl hydrocarbon receptor. Third receptor was LXR receptor. And the same for 2023 dihydroxy, which is a different vitamin D hydroxy derivatives. The number one receptor was aryl hydrocarbon receptor, and the VDR was the second receptor. This really led us, this bioinformatics analysis by David Grossman led us to computer modeling, which showed the really good the mass bind receptor, good docking stall, functional analysis, which showed that they really interact with the receptor, with 2023 dihydroxyvitamin D3 being the best one, and then downstream transcriptional activity of the genes which were downstream, showing that it's unquestionable the novel vitamin D derivatives act on the aryl hydrocarbon receptor. This has been confirmed most recently by the paper by Yue Song as the first author. We have additional paper just recently published, which confirm again the CYP11A1 vitamin D hydroxy derivatives, as well as classical ones, they can act on the aryl hydrocarbon receptor. Then we moved to test LXR, liver receptor alpha and beta, because it was coming on the microarray, it was coming in the IPA pathway. So we use commercially available kits to all of them act as agonists on LX alpha or LX alpha beta. There is certain specificity, some as agonists with different affinity, some as reverse agonists. Then it was by immunofluorescence translocation into from the cytoplasm to the nuclear ligand induced translocation. This is the quantification. This is ethanol control induction translocation. It was Alan Oak done. Then here it was done by Radomir Slominski using the flow cytometry, image flow cytometry, shows the ligands, they induce the translocation of the LXR alpha and beta into the nucleus, just demonstrating that and then there was a computational modeling, not enough time to talk about this, but it was done by UAE Song under the supervision of Yuhua Song, which further confirmed and simulation on the interaction of Luminosity with the LXR receptor. So we are coming into the end. What is the, for computational biology? A lot, because there is a lot of the at least 15 hydroxy derivatives of vitamin D. And the L3 lumisterol, I focus only on both of them. Vitamin D3 is vitamin D3. They enter the cell, enter mitochondrion, because CYP11A1 is mitochondrial enzyme and is hydroxylated, different position, and which are further hydroxylated by other enzymes, some of them microsomal. So we have large number of hydroxy derivatives, and this is a classical one. So let's look. Different colors, and they interact with the different receptors. Three of them, they interact with the aryl hydrocarbon receptor. They can interact with the retinic orphan acid receptor alpha and gamma, and to carry the transcriptional activity. Some of them, like vitamin D and 125, they interact with the genomic binding site of vitamin D receptor, because vitamin D receptor has two sites, non-genomic and genomic binding site, complex with RXR, regulation, and phenotypic activities. Then there is a binding, and unfortunately not vitamin D, but 125 and lumisterol hydroxy derivative, they can bind to non-genomic binding site, and which are membrane bond transmissions. This is a whole concept of the nuclear receptor acting on the membrane and signal transduction regulation of function, or perhaps even interaction with RXR, but this is under the question mark, and transcriptional activities. So, and then we have here what we have discovered, the new LXR receptor as two of them. So we have several molecules, several receptors, different phenotypic effects. So just to come to the conclusion that novel corticosteroids and lumisterol compounds are produced in the body. They are excellent candidates for treatment of hyperproliferative and inflammatory disorders as an anti-cancer and agent and radioprotectors. And here I would like to show the team at the different times. This is at UAB, it's Robert Taki, he's expert in CYP11A1. So students, and here is the team which was at University of Tennessee working, and then acknowledgement, the group of the people who really were interact. In red are all of the people who are working on this exciting project with multiple implications. So in my laboratory, Chander, Atar, Jack Chen started working with us on the bioinformatic part, UA Song, Yuhua Song, and these are the different NIH grants, VA grants, which really have supported and are supporting this project. So I will stop here and we'll wait for the question. If there are questions, depending on the question, I have few additional slides to show.
Thank you, Dr. Slominski. Any questions from the audience? So we're just on time. A very quick question. I'm totally out of this scope and not in this field. So it's a naive question. So the vitamin D, I noticed that there's a huge change in terms of gene expression, right? Profiles and so do you know it target, like if there's some epigenetic changes, is methylation or how vitamin D works?
Multiple. It is very pleiotropic effect that so far the dogma was one molecule, 125, one receptor, vitamin D receptor. But there are multiple effects, and sometimes there are contradictory biological effects, and it is known for decades. So it is very difficult was and to explain how it is possible. The same molecules has the different activity and acting through the same receptor. And now we have explanation. There are many more molecules. You take the vitamin D, or it is produced in the skin through the sun, and it is depending whether it will access adrenal gland, depending on the enzymatic composition, you have different activation. Your question is also very good, because when you take orally vitamin D, so it is, anybody who knows physiology first will go GI and go to the liver, and it will be hydroxylated in 25 position, and this is a classical pathway. And 25 hydroxy, which I didn't say, it is not a substrate for CYP11A1. CYP11A1 acts only on the vitamin D3 and hydroxylate in the different positions. So it is the same structure, only the hydroxyl groups are on different position. And this addition of this hydroxyl groups to different position changed the specificity for the different receptors. Either it is VDR, either it is LXR, either it is AHR. And in fact, there are other transcriptional regulators which are being detected. I have sent recently half a milligram for 5 compounds to Anton Jeter, to NIH, because they have the so-called screening for nuclear receptors, like for 50 nuclear receptors at once. And it is coming unbelievable, because small change in one hydroxy group change the specificity. So this is an open, completely open area. And we know that thousands of genes are really regulated by vitamin D, but it's vitamin D downstream metabolites of vitamin D with different receptors at different body sites.
Okay, got you. Thank you. Thank you so much, Dr. Slominski. So we are running out of time. So our next speaker is Dr. Claudia Carvalho. So our close friend. So great to see you here, Claudia. So you probably can start to share your screen.
Thank you, Zenchen. Let me see.
And Claudia is an assistant investigator of Pacific Northwest Research Institute. She's also affiliated to Washington University in Seattle, if I recall. Yeah.
Yes.
It's your time. Thank you.
Thank you, Zenchen. Thank you so much for the invitation to share our work here. So my lab, as Andrzej said, my lab just started actually 1.5 years ago in Seattle. So we are hiring, if somebody is interested in studying rare diseases and structural variation in rare disease, please contact me. Oh, it's not, sorry, something happened here. Let me stop sharing, try again, Zenchen, sorry.
Sure.
I don't know why. Can you see it?
Yeah,
Let's see. Let's see if, I don't know why it's not passing. I'm not sure. Oh my gosh, sorry.
It looks well from my end.
It looks low?
No, it looks great from my end.
Yeah, I cannot change the slides. That's the
okay.
problem. And I don't know why. Let's stop sharing again. Let me try the normal mode without the presentation to see if it works. Do you see my slide?
Yes, I can see it.
Because then I can change, sorry. So my lab study structure variation in the human genome, and in the different types of copy number gain, copy number losses, inversions, and complex rearrangements, which are actually can be a mix of duplications and deletions and inversions, and they are very challenging to be detected and studied depending on the technology we are using. So our work is very towards the study of rare diseases and constitutional diseases. So The research goals in my lab are: 1, to define the best technologies to detect and resolve pathogenic structure variants; 2, to investigate their molecular features, origin, and the mechanism of formation; and 3, to explore the impact of them in rare diseases, including phenotypic variability and incomplete penetrance. So one very important feature of the human genome that has a strong impact in the constitutional diseases is the fact that 50% of the human genome is constituted by repeats. So several of them are important to lead to variants that cause disease, but one is especially important, and it's called segmental duplication. Segmental duplications, they can vary in size about 1 to 300 kb. They affect all the different chromosomes with different, some chromosomes have more segmental duplications than others. And the recent T2T reference indicated that segmental duplications constitute about 7% of the human genome. So in fact, structure variants in rare diseases, they often map nearby or at the segmental duplications. And here is a map of several of the disease that we already know, and if they are defined by deletion or gains, and the presence of segmental duplications. As you can see, they map very close or on top of them. And so repeats are actually excellent predictors or where the structure variants, the de novo structure variants are going to happen and which ones are going to cause disease. This is a very important point because to study rare diseases, we need to be able to detect them. And here I'm showing you an example of one type of variant that's considered simple. So by read-dep, we were able to detect a copy number gain involving chromosome X. This is a pathogenic one. And studying the split reads, we can easily define the structure of this variant, which was a simple tandem duplication. The breakpoint was easily detected by split reads. However, another patient had apparently simple duplication as well. However, when we investigate the breakpoints, one of the breaks was easily detected by, we could see easily by split reads. However, the second breakpoint, it mapped to segmental duplication. In this case, is a segmental duplication involving opsin genes that are in the reference, they are in 3 copies, very similar to each other, about 99.9% similar. And most of the variant callers, they do not detect, or I mean, they detect, but they do not report this type of variant. So if someone is using only split reads to detect the structure variant, not copy number gain or read-dep, then this variant is not going to be detected. The other problem is that even if we can detect and use read-dep information, it's we cannot completely resolve the structure because the junction inside the low-copy repeats are not helpful in using short reads. We cannot really define which one of the opsins was actually involved in the rearrangement using only the information in the short reads. So another problem is that inversions and complex rearrangements are still a very challenge to be identified. Many times they are called by, they are false positives and false negatives in the genome. And one of the reasons is the fact that many times they involve repeats as well, and they have more than one breakpoint junction. That's the definition of a complex rearrangement. So 2 types of complex rearrangement that we see very often in rare diseases is the triplications that are flanked by duplications, and the duplications that are interspaced and separated by a region of copy number. So both of these types of structure variants, they actually have 2 breakpoint junctions that are in cis. So for someone to be able to detect them, and like without knowing the read-dep, without knowing the CGH, for example, as I'm seeing here, you should be able to detect both of the junctions in cis at the same time. So here is an example of the problem that we see with this type of complex rearrangements. For example, for the inverted triplication shown in this array, one of the junctions maps to unique regions, and the split reads can easily detect and resolve. However, the second junction maps to inverted repeats or segmental duplications that are in inverted orientation, and the short reads do not see them at all. So our hypothesis is that using long reads and optical mapping in these types of patients, we will be able to detect and will be able to resolve pathogenic structure variants that have multiple breakpoints in cis. To study and to study our hypothesis and to test it, we are enrolling patients with a syndrome called MACP2 duplication syndrome, which is an X-linked neurodevelopmental disorder that is caused by copy number gains of the MACP2 CpG binding protein 2. This is a protein that is a transcriptional regulator that is essential for the normal function of the central nervous system, affects mainly boys, and accounts for 1 or 2% of all cases of X-linked intellectual disability. Why we decided to study these patients is because it was already shown that complex rearrangements contribute to about 20 to 30% of the patients with this disease. So to study this cohort and to test our hypothesis, we are applying different assays. We have the copy number assays that we use in our lab, which are customized arrays, droplet digital PCR, sequencing like short reads, long reads, PacBio, and nanopore, as well as optical mapping. So for all of the patients, we are using all these technologies to study them. So this is how our cohort looks like right now. This is a preliminary data that we are still doing a lot of the work, but we have now 86 patients that we already collected, and all of them already have customized arrays. So we know that half of them have apparently simple duplications that are we still need to sequence to make sure they are simple. And we have 23% of patients with complex rearrangements that I mentioned before, triplications flanked by duplications and interspecies duplications. This is confirms previous studies showing the same, about the same range. And we have 20% of these patients with terminal duplications. This was a surprise for us. We didn't see this type of variant in so high frequency before in this cohort. And what terminal duplications are, they are actually duplications that involve the MACP2 and they go until the telomere. So they are pretty big, about 2 megabase to about 16 megabase. And compared to the other duplications that involve the same cohort, the other duplications are about 506 kb in average. So they are much bigger and they carry many more genes. So they may be contributing to the different phenotypes, variability in phenotypes that we see in this cohort. So it turned out that the terminal duplications are pretty diverse types of duplications, and they are actually complex rearrangement for most part. So one type of terminal dupes that we see in this cohort are recombinant X chromosomes, which constitute 30% of the cohort. And the recombinant chromosome that actually are constituted by the duplications involving MACP2 and the deletion involving the XP region. The unbalanced translocations are 53% of all the terminal dupes that we have, and they actually involve duplication in the XQ region and the deletion of another chromosome. So for the simple types of terminal duplication, still complex, but simple in the sense that they are easily to resolve, short reads were very easily, could detect the duplications and the deletions in the same chromosome, and we could detect the junctions by split reads. Optical mapping was used as an orthogonal methodology and shown the fusion inside chromosome X and the junction between XP and XQ. And the structure was resolved as expected. For some of the unbalanced translocations, we also could resolve by using all these methodologies, both short reads, read depth, and the split reads, and the optical mapping showed the translocation between X and Y, and we could easily resolve this case as well. However, for 50% of the terminal duplications, we could not resolve so straightforward, and not all the technologies helped. For instance, in these two cases here, the terminal duplications were mediated by the segmental duplications. So in this case, inverted repeats, and the same inverted repeats actually caused another terminal duplication that's consisted by a triplication followed by a duplication. So despite the inverted repeats are the same, the structures are different. In each of these cases, the results were different as well. So for this patient with the triplication, optical mapping was actually the technology that really helped resolving. You can see here that this terminal dupe is actually a translocation between X and chromosome 7. And the interesting thing about optical mapping is that we could see both junctions of this complex rearrangement in cis in the same molecule. So we saw the triplication and the translocation to 7 in the same molecule. And using this information, we actually went to the short reads and the long reads, and the only one that really could show the junction in terms of nucleotides was nanopore. But we had to use the optical mapping to actually find the junction in IgV. So the other case that also had the breakpoint mapping to the inverted repeat is this BAB 12077, and actually optical mapping showed that this case is actually an inverted duplication involved a huge stream 0.1 megabase of an inverted duplication and carrying two junctions, as usually we do have for all these complex rearrangements. Short reads could not detect at all, and nanopore and PacBio were able to resolve. In this case, we use optical mapping to resolve, and also Sniffles 2 that we have been working together with Fritz Sedlec group. So in all, our work so far, we observed that from the 86 patients that have duplications involving MACP2, actually half of them have complex rearrangements. They are not in tandem duplications as usually we think about copy number gains. And we had to use multiple technologies to be able to resolve all these complex rearrangements and to see how the structure for every individual patient. In summary, structural complexity is a feature of genomic rearrangements at this region in the XQ, but we also expect this to be a feature of other diseases that affect chromosome X, because the presence of inverted repeats is pretty high on this chromosome. Molecular characterization revealed that complex genomic rearrangements occur in almost half of this cohort. Inversions often accompany these complexities, and the inverted repeats stimulate or mediate formation of complex rearrangement. We also observed that combining technologies is very important for studying structure variation in constitutional diseases and to discover and detect and resolve complex genomic structure. With that, I would like to first say sorry for the problem with the slides. And thank you, my patients and families, my institute, and my lab, Mira, Michelle, and Kelly, who has been doing great work here, Chris and Howie, who have been working from Baylor together with us very closely, Davu, who is the neurologist working with us, and Fritz Sedlacek and team, who has been working also to help developing some of the tools that we are using in this project. Thank you.
Thank you, Claudia. This is awesome, as always. Great job. So any questions from the audience? So I don't know the optical mapping approach is so powerful. I was thinking the resolution is limited. So what's the evolution, or what's the resolution for the optical mapping in the if it's not base resolution, right? It's based on some enzymes and so it's quite large scale, if I recall.
Yes, yes. So the good thing about optical mapping is that there is like the molecules can be pretty big. So we have some cases that we have like 500 kb, sometimes 800 kb. And the good thing is that we don't get nucleotide information in terms of like the what's really going on with the junction per se. But the good thing is that we can see the junctions in cis. So these cases are all like de novo, right? Most of them are de novo, or they're super ultra rare. So usually you don't know, is this an alteration in the reference or the parent that has already this alteration in another chromosome? So it's important to see that the multiple junctions that we see there are in the same molecule. So you are right, the resolution is not super high. It's about one label per 7 kb more or less. But if we use the information of the optical mapping together with the short reads or the long reads, we get a very powerful tool to find the junctions. Because as I showed, some of the inverted repeats or the segmental duplications, even long reads like nanopore cannot. They cross, but we cannot distinguish them inside. So because of the similarity is so big, but because of the size of the long reads, sorry, the optical mapping, we can see the junctions there. Even we don't know the precise junction, but we see they happening there.
Gotcha. Another question is, is still targeted sequencing like the Beck et al., the cell papers that we worked previously, or it's the whole genome sequencing for the long reads?
This is all whole genome sequencing.
All whole genome sequencing. Okay.
We have PacBio-Hi-Fi and nanopore all for all whole genome.
Okay. It's expensive, right? If a whole genome instead of a target, the probe is expensive, right? If I recall.
Yes, yes. So PacBio is the most expensive of all of those, but the price has been decreasing with time. Nanopore is about 2 times short reads. Yeah, so it's still some, expensive, but to resolve this type of alterations, I think we have to learn first and then make an automatic way, right? In the future, that's the goal. But first we need to learn we need to learn what we don't know.
Sure.
That's the goal.
Awesome. Thank you so much.
You.
So our next speaker is Dr. Leng Han from Texas A&M. He's an associate professor, and he will talk about big data for precision oncology. So Leng, it's your time. You're muted.
Okay. Yeah, can you hear me now?
Yeah, can hear.
Yeah. Thank you so much for the invitation, and it's really a pleasure to meet many old friends and new friends. So today I'm talking about this harnessing big data for precision oncology. So basically, there's a lot of different big data resources. For example, we summarize here in a recent perspective in Nature Review Clinical Oncology. Basically, the first one is the clinical studies, like those clinical trials, which we can do a meta-analysis to identify some of the targets, make some clinical decisions. And then the second one is the real-world data. Is it's kind of like health record. So basically, these provide another dimension for the data resources. The last piece is what we are quite familiar with, the omics data. For example, the TCGA data resources, the cancer genome ATTIS data resources. They have like more than 10,000 patient samples. Each of the samples, they have a different kind of clinical information and all kinds of omics data like DNA sequencing, RNA sequencing, DNA methylation, etc. So basically, for each of this kind of data resources, they have their advantages and disadvantages. For example, the clinical data they obtain in a very controlled clinical settings, so they can directly relate to the clinical settings. However, that many of the key patient level information, such as the sex, treatment history, are kind of not published. It's very difficult to obtain all the related information. The real-world data is kind of information from the tremendous large number of patients from the like electronic health record, And however, the quality and the completeness of the real-world data can be suboptimal because this is, real-world data. Sometimes the physician scientists submit those information. Sometimes the patient themselves can submit the information. Multi-omics data is can provide novel insights into the molecular mechanism. But at least in the past several years, most of the large consortium, they do not include those clinical information, especially for the treatment information. That's very rare, especially immunotherapy for those. But I think in recent years, this pattern has been improved a lot. So there's a lot of large-scale cohort with the immunotherapy treatment for this kind of omics data. So today I'm mainly focused on this kind of immunotherapy. We know that the anti-PD-1, anti-CTLA-4 immunotherapy has kind of revolutionized the cancer treatment. Many of the patients benefit a lot from the immunotherapy. However, you know that as long as the treatment, they also have some toxicities, right? So basically, this is a kind of balance between the efficacy and the toxicity. In the past several years, our groups kind of focus on both sides of the immunotherapy. For example, we focus on how to improve the efficacy, how to predict better biomarkers for efficacy. On the other hand, we also did a lot of analysis for the toxicity, try to understand what is going on for the so-called immune-related adverse events. So the first topic I want to talk about today is about this for the immune checkpoint inhibitors. What is the results difference between the male and the female patients? So you know that there's a lot of awareness for this kind of anti-cancer drug treatment between male and female patients, and there's a lot of great reviews summarize the difference between male and female patients. For example, for autoimmune diseases, infectious diseases, etc. And the previous researchers think that the hormone play very important roles, but recent studies also show that many other factors also play important roles driving this difference between male and female patients. So back to 2018, there's like 2 papers, one published in Lancet Oncology and then another published in JAMA Oncology. The Lancet Oncology paper concludes that there's the male patients benefit a little bit more than female patients regarding to the immunotherapy. And a few months later, another paper published in JAMA Oncology, that's the below ones, they show that there's almost no difference between the male and female patients. So this is kind of kind of controversial data. They both use the collect the clinical trial data. They do the meta-analysis. The approach is quite similar, but why do they obtain quite opposite results or say conclusion? So here we go to the digging deeper for this data. So basically, this is the summarize for the data set they used in their study. The upper one is the Lancet Oncology paper. The bottom one, this one is the JAMA Oncology paper. And you can see that for each of the data sets, although they do not reach the significant, significant p-value, but they have some bias towards to a certain male or female patients. And you can see that we highlight this in the blue cohort. Those are kind of biased a little bit in the male patients. And this pink panel shows that these patients may have a slightly better odds ratio than in a female patient than male patients. And you can see that these 2 studies actually collected different clinical data. So basically, the first one, they collected more male biased patient samples. So they conclude that overall they can get a significant p-value for overall together for male patients. However, the JAMA Oncology paper collected some more female biased patient cohort, so they can conclude that there's almost no difference between the male and female patients. So here we think that that of course they have their own criteria to select the samples, select the patient cohort, but we think that the omics data will help a lot try to understand what's going on between the male and female patients. So here basically we use a very rigorous statistical analysis called the propensity score, try to control the confounding factors between the male and female patients. For example, the smoking history, the age, the race, and the state, etc. We compare all those known potential biomarkers for immunotherapy. For example, the tumor mutation burden, the neoantigen, the TCR, BCR, checkpoint, immune cell abundance, and copy number variation, etc. And here you can see that on the y-axis is different cancer types. On x-axis is a very key, the potential key biomarkers for immunotherapy. you can see that the lung cancer has a quite a higher level of these features in the female patients, plotted in the red. And while those like melanoma and kidney cancer, they have more biased female pattern, that they have a higher level in the female. So you can see that for those meta-analysis, that several years ago, the majority of the clinical trials are coming from either lung cancer or the melanoma. And here you can see that these two cancer types actually show very opposite pattern for the omics data. So you can imagine that if you pull these two different cancer types together, the conclusion may not be robust or say reliable. Okay. So furthermore, we'll compare those like immune cell abundance. This is like a very key factors for the immunotherapy, like the CD4 T cells, CD8 T cells. And you can see that again in the lung cancer, it's kind of showed a very strong bias towards the female patients, while in the melanoma, it's kind of more like a male bias. And for all different kind of checkpoint, we see the similar pattern that in the lung cancer is kind of female bias, and in the melanoma patient is kind of like a male bias. So the conclusion from these studies that one is that we probably should not put different cancer types of patients together for meta-analysis, which will not be robust and reliable. Another one is that we think that omics data with the clinical information is critical for us to further understand what's going on between the male and female patients. This work has been led by our outstanding postdoc, former postdoc, Dr. Ye. Now is a PI at Shanghai Jiao Tong University. So the next part, we're trying to understand what is going on with the immune-related adverse events. As I mentioned, that this is a toxicity for the immunotherapy. So basically, when the patient has been treated with the immunotherapy, you know that because it will activate the immune system in the patient body, it definitely will cause some of the toxicity. For example, the myocarditis is kind of affecting the heart. The occurrence frequency of the myocarditis is not that high. It's like maybe less than 10%, but the death rate for this kind of RE is very high. It's like 50%. So it's very, very important to study this kind of potential immune-related adverse events for so-called for the risk for the patient. Try to control these, try to manage the patient. So far, there's not a good biomarker for RE, at least for the tumor samples. That there's a lot of studies trying to using the blood samples to get some potential biomarkers for RE. I think one of the difficult issues is that it's very difficult to obtain the RE samples. It's not like a tumor samples. It's kind of affecting those normal organs or tissues. It's very difficult to get those samples, right? It's not like tumor samples you can cut as well as you want. So basically, we use a very alternative strategy. Here, we obtain the reporting odds ratio for the REs from a database named the FIRS. It's FDA approved adverse events reporting system. Is what we call belong to the real world data part. So in this database, in this data portal, there are more than 50,000 adverse events recorded and covered more than 18,000 patients, spanning over 26 cancer types. You can see that this is a large number of patients recorded here. It's much larger than any of the individual patient cohort. So we also use the TCGA data to calculate the different kind of molecular features for each of the cancer type, and then correlate these molecular features with the odds ratio across different cancer types. So here's one example. You can see that we can use a linear regression model to predict to see whether the molecular features can predict the RE odds ratio. So basically, you can see that this is a good prediction. Like RS is good. It's almost about 0.75. So this is just a known biomarkers for immune system. So we try to expand this to a global pattern. For example, we screen every pairs of genes, and we can see that there's we screen 2 genes has showed high prediction power. This LCP1 and ADPGK, the prediction power can be as high as 0.91. And we also get some samples like with the RE or without the RE. This is again, the sample is very difficult to get. So we obtain 28 samples together, 14 patients with the RE, 14 patients without the RE. And you can see that we use the antibody staining definitely show that the LCP1 and ADPGK show high expression in the patients with the RE. We then build an AUC prediction model to see that whether these two genes can predict the RE occurrence. So basically, we think this because these two genes kind of involved in T cell activations kind of makes sense, right? Because once the T cell has been activated, you are very more likely to attack the normal tissues to cause this kind of RE. So this paper has been published in Nature Communications. So we further collected different data, try to understand what is the associations between the REs and ICI efficacy. And this has been studied a lot. So basically, this is a perspective we summarize here. Different studies use different strategies. For example, majority of them use individual clinical trials. They can compare whether those patients has, REs has a better efficacy while receiving the ICI treatment. So basically, the lower ones, they use different strategies. They can use the meta-analysis. And the last one, I think they even use the kind of different multi-source data here. So, based on different data source, they can have different conclusions, suggesting that there's necessary to further study what's going on between the RE and ICI efficacy. Similar to this, we also summarize the associations between the REs and the demographic factors. For example, when we compare young patient and old patient, we can see that there's some difference for the REs for certain type of REs. And also when we compare male and female patients, we can also see quite difference. For some of the studies, they show that there's a female bias. For some of the studies, they show there's a male bias. And also this is also rely on what kind of immune-related adverse events we're talking about. So this is still ongoing directions, and there's a need for a lot of effort to try to understand what's going on for this kind of what kind of key factors will affect the RE. So this is the RE work mainly led by Dr. Yin Jin. Now he's also PI at Shanghai Jiaotong University, and the review paper is also led by both of them. Jing Wen just joined my lab a few months ago. She contributed a lot for this review paper. So other than the immunotherapy part, our group also interested in different kind of innovative therapeutic approaches. For example, in the past several years, we using large-scale molecular data as well as those functional screening data, drug screening data, try to understand the clonal therapy. For example, is there if we can find a better treatment time point to treat the patients, which can get a better response. Like hypoxia target therapy, we try to understand whether we can utilize the hypoxia status to achieve a better treatment efficacy for the patients. And many other studies published in the past several years. And other than these, we are also trying to identify novel therapeutic targets. One of the major focus from my lab is that we try to focus on one is a non-coding RNA, the other is a post-transcription regulation. So non-coding including not only link RNA, but also pseudogene enhancer RNA, SNL RNA and circle RNA, and post-transcription regulation, including like alternative splicing, RNA editing, and alternative polymorphisation. So regarding to these, we published a series of cancer genomics paper, and this is a summary for what we have done in the past several years. So finally, I would like to thank for all my professor lab members, and many of them, some of them have already left the lab and started their own labs in the prestigious institutions. And also thank you for the funding support, as well as many of the collaborators that help us to perform the experimental validation, provide the clinical insight and biological insight like that. Also, my lab is recruiting for like all levels of personnel. Thank you so much.
Thank you, Leng. This is awesome. This is impressive. Leng is a star scientist in Texas, I would say. So any questions from the audience? Probably people are tired a little bit. Yeah, I have a quick question regarding the TCGA actually has no anti-PD-1, no therapeutic data at all, right? So all of these are kind of a prediction, right? You have to use additional data to validate your discovery. Is that true?
Yes, that's true. I think the immunotherapy is not very popular back to when TCGA started. TCGA started like more than 10 years ago.
Yeah.
But I think that the good thing is that, as I mentioned, there's many more studies they have using anti-PD-1. They have the omics data. It's just a little bit difficult to assess. You need to try to apply for those sets. But there's many of the data sets already there. So yeah.
Another question is, I think if I recall, the anti-PD-L1 or PD-1 therapies, it's more effective in specific cancer types, right? Like lung or melanoma, this type. So the data for some other cancer types are limited, right? This is another issue for the anti-PD-1 data analysis, right?
Yeah. As you mentioned, that melanoma definitely, I think, is the first one using this anti-PD-1, and then lung cancer has a lot. So the most of the the validation data set is coming from melanoma or lung cancer. That's true, but I think there's more data is coming out, like prostate cancer. You can find a limited, yeah. But again, I mean, for pan cancer analysis, as long as you can find some independent data set for validation, that would be great, right? So yeah.
Awesome. Okay, thank you again, and we will switch gear to the next speaker.
Sure. Thank you.
Thank you. Maisie Zhou. Maisie, yeah, probably you can share your screen. So Maisie is the assistant professor from Vanderbilt University. So I think she started her lab just last year, right? Oh, 2 years ago.
2020, around the pandemic.
Okay, yeah, it's your time. Go ahead.
Okay, great. I'm delighted and honored to be invited to speak today. So, let's see. Okay, so here's the outline of my talk. So first, I will give some background for the research, and then I will go through the first tool we have developed, Aquila. So basically, it's a diploid sampling and variant detection algorithm used in increase. And then I will spend a little bit of time to go through some other tools developed in the lab. So today, how we actually sequencing human genomes. In 90% of the wet lab, we're still using short read sequencing, which, for example, in the wet lab, we have DNA copies, and these DNA are cut into short DNA fragment, usually around 500 base pair. And then this short fragment will be sent to Illumina sequencer to be sequenced to generate billions of short pair reads. And this reads usually around 150 base pair. So users can use some alignment based algorithm to align those reads against a human reference to detect variations. So here's a screenshot visualization from after we align the reads against the human reference, how we actually detect variation. So for example, here is our human reference, and here is at this position, we can see here the reference allele is G, and we see a lot of reads show A here. So A is the alternate allele, and here reference allele is A, and we see a lot of reads show G here. So G is the alternate allele. So today, we have discovered several types of variants in human genomes. For example, they classify to small variants or structural variants. For small variants, usually less than 50 base pair, including, for example, SLEEP or SMV, and small deletions, small insertions, or small repeat variations. And for large variation, usually larger or equal than 50 base pair, we call them structural variants. But compared to the reference genome, we know we have duplications, deletions, inversions, and translocations. And duplication and deletion are often called copy number variation, CNV. So these are widely studied in cancer genomes. So after we detect the variation, based on the sequencing reads, this is also called variant genotyping. But there's another procedure is also very important, which we call phasing. So basically, try to determine which combination of like variants from the which variants are from the same haplotype or which are from the different haplotype. For example, let's use this slide again. So we can see here. this bunch of reads actually, we can see A and G, these two autolated allele are showing the same, this bunch of reads. We know this A and G are from the same haplotype. But you could think about in the real scenario, this G could be really far away from A. Then if we use short reads, it's actually hard to know this AG from the same haplotype or from different haplotype. Okay, so today we have several different types of genome-wide sequencing technology. For example, like what we introduced, short reads. So for short reads, the sequence error is low and it's very cheap. But the disadvantage, because it is short, it's hard to detect large structural variants and to phase the variants. Today, we also have link reads, which we often call the barcoded short reads. So for this link reads, basically, you can think about it as short reads attached by a barcode. Then we know the reads with the same barcode originating from the same long DNA fragment. Then they provide this long-range information for us to easily to reconstruct the diploid genome and detect and phase structural variants. And because in the use short reads, so basically inherit all the advantage of short reads. But today, because the disadvantage today actually 10x genomics discontinues the 10x link reads because some patent issue. But still, today we still have like China BGI and universal sequencing companies still provide these promising link reads we can use. And of course, today we also have a lot of long reads basically can provide us average like 10 kilobytes long reads. And for this long read sequencing, for example, PacBio long read sequencing, basically the advantage is still we can use them to reconstruct diploid genomes and easy to call and phase large structures. But the disadvantage, because compared to short reads, usually the sequencing error is high and it's more expensive and it require stringent input requirements. So my lab basically use all different short reads, link reads, and long reads, different sequencing technology to build architecture to solve several problems. So first, how to actually generate personal diploid genome sampling, and then how we actually detect sleep, small indels, and large structural variants. And then how, after we do the genotype, how we actually phasing all the type of variants. But the long term right now, the long term goal in the lab basically how we because with as we know with the cost effective sequencing different sequencing technology and the available abundance of the whole genome sequencing reads. So my the long term goal in the lab basically how to develop a large scale machine learning based algorithm for large scale population genomic data. Okay, so first for today's talk, I actually want, first the tools I want to introduce is Aquila. So basically, Aquila is a human reference assisted local de novo sampling algorithm using 10x link reads. So, here's the overview of Aquila. So the input data for these tools basically is a high quality reference genome, and then your whole genome sequencing, for example, link reads. And we have several modules for these tools. So let's start the first module. Basically, we call the haplotyping module. So basically, it's how we actually partition the link reads to different haplotype. Okay, so to do that, we first need to reconstruct and annotate the long DNA fragment. So because for link reads, basically barcode reads, we know reads with the same barcode came from the original long DNA fragment. So we can, based on this idea, we can reconstruct this virtual long DNA fragment and then annotate them with the heterozygous sleeps. For example, here we can see the autolated allele is G, and we see this reads show G here. So G is the same as autolated allele. We allocate one is for autolated allele. And here we can see the reads show C here, and C is the same as reference allele. So we allocate with 0. 0 is for reference allele. So we reconstruct all the long DNA fragment based on the idea reads with the same barcode. Basically, we reconstruct a lot of billions of these long DNA fragments. And then we allocate them with the heterozygous sleep. So why we want to allocate them with heterozygous sleep? So the basic idea is this. For heterozygous position, if we know the long DNA fragment from the maternal haplotype, if they show 1. So we know the fragment from paternal haplotype, they show 0. So, but at this stage, we don't know the fragment is from maternal or from paternal haplotype. That's what we want to figure out. So let's take a pair of heterozygous slip as an example to illustrate how we use our model to do this. So for this pair of heterozygous slip, we know the real scenario, the real haplotype is 0011. And after we allocate all these long DNA fragments with the heterozygous slip, what we will observe is we will see a lot of long DNA fragment support 00. We put in the 00 clusters, and a lot of long DNA fragment support 11. We put them in the 11 clusters. But we still see a few DNA fragments support 0110, we put here. So what does that tell us? So basically, we observe this pattern. So we see this, we see a lot of long DNA fragments support, give us supporting power. This even parity, this 0011 haplotype is correct, but the other odd parity actually is wrong. Because they caused by sequence error from the reads, and that's the scenario we need to exclude. So here we involve a Bayes model to determine which given this observation, what's the correct haplotype. Okay, so after we, for each pair of these heterozygous slip, we basically, after we involve our Bayes model, basically we exclude those two clusters. Suppose the wrong haplotype, only keep these two clusters, suppose a correct haplotype. Now, and we end up with billions of these pair of small pairs, clusters. And now what we can do is we can involve a recursive algorithm, basically can collect all the clusters, support the maternal haplotype in one direction, and then in the other direction, we can directly collect those clusters, suppose the parental haplotype. So eventually we can end up with thousands of these phase blocks, and within each phase block, we know we have two big clusters. Basically, each cluster, we already partition those long DNA fragments, those linkers to different haplotypes. Now we basically finish our first modules. Okay, so let's talk about our second local sampling module and the contiguity module. So basically, after we end up with thousands of these phase blocks, for example, this phase block, we have two clusters, and within cluster, basically now we have the reads, we can do a sampling. But because at this time, after we have all these phase blocks, the maximum phase block with this link reads maximum can reach 100 megabytes. That's around like a chromosome size. So we don't want to assemble all those reads in one big chunks. What we do is we involve this local sampling strategy. So basically the idea is for each phase blocks now, we can involve a high confidence boundary point. We can cut each phase block to small chunks. So this is a user free parameter. So for example, can cut to 100 kilobase chunks. Now for this phase block around 400 kilobase, we end up with 8 small chunks. For 4 small chunks for each haplotype. Now, what we can do is we can take a small chunk of these reads. Now we can perform a de novo sampling to assemble those reads together to achieve the full-length contiguous sequence, which we call actually mini contiguous sequence, mini contigs. For example, here now we achieve these mini contigs in each these small chunks independently for each haplotype. Now, because based on this high confidence boundary point, we can actually concatenate now together in the contiguity module to achieve our final full-length contiguity sequence, the contigs, which we called. So let's go to the last module, variation module. So now because, not like the alignment algorithm, now because we assemble the reads together and achieve our full-length contigs, now we can just do a simple pairwise alignment from the contigs against the human reference to detect the genome-wide variation. And because our sampling is already haplo resolved sampling, so we actually can phase in all the variations. Okay, so in the paper we did a lot of benchmarks, compare a lot of use a lot of different libraries, sequencing libraries, and compare different tools. For example, sampling tools, variation tools, and phasing tools. And basically, what we show is compared to short reads, which can do well on sleep and small indels, Aquila can do well on not just small variation, we can do good SV indels, and we can generate these diploid genomes. We can do very good breakpoint precision. We can provide user full phasing sleep small indels and SVs. Okay, I think I still have a little bit of time to go through some other Aquila related tools developed in the lab. So the first tool is called Aquila STFR. So basically, as I mentioned, so China BGI invented this promising single tube long fragment read, which called STFR increase. So basically, we extend Aquila to this STFR increase. Now we also provide a module you can using both can do hybrid sampling for both increase and the STFR TANS increase and STFR increase. And we also have another tool called Aquila SV. So this is a region-based diploid sampling and variant detection tool. So the basic idea is because the whole genome diploid sampling usually is computationally intensive. So when researchers usually, if they're only interested in some target region and maybe they want to do some population studies, then we provide the tool which you can do target region-based diploid sampling, and you can use this sampling to cause a variant detection, and it's much efficient for population studies. And so another tool we introduce here is Aquila Deep Filter. So because no matter what the variant detection tools from short reads or from linked reads like Aquila, or other tools from other variant call from short read, we often have some false positives. So how to actually not sacrifice the sensitivity of the variants call, but still increase our precision, basically decrease all, basically just remove all those false positive calls. We introduce this example deep learning framework, basically can refine the large deletions in both in both short reads and linked reads. Okay, so the last tools I actually want to introduce is actually I talk about the long-term goal in the lab. Basically, because we have available abundance of whole genome sequencing data, population scale data. So how to actually detect the large structural variant? How to design a efficient, like a precise, better SV caller from population data. So this is actually first tool we propose. So here the basic idea is instead of usually we rely on alignment-based algorithm call the individual independently and then merge the SV breakpoint through some hard threshold, which we often have bias. So here we use diploid sampling. For example, here we use in this study we use 30 individual hybrid sampling from linked reads, and each person basically we have this diploid sampling, and we involve this multi-sequence alignment, and we also involve some ape sequencing, for example, chimpanzee, orangutan, and macaque sequencing. We can better. And then we can actually rely on this alignment-based algorithm, multi-sequence alignment-based algorithm basically can better to detect the S3 based on the population from the global view with the population data. Okay, so here I want to introduce some of my collaboration. So my lab basically is computational lab, so we develop algorithm, but I have some interest in neurology disease, for example, autism. I have some collaboration from Vanderbilt Medical School, for example, Jim. And my lab also interested in some neuroscience studies. We use deep learning, for example, recurrent neural network to model the brain activity, which we have some collaboration with Christos. And so my lab also develops, accept the whole genome sequencing data, and we also develop algorithm, for example, how to detect CMV from single cell RNA-seq, single cell DNA-seq, or right now, since 2020, we have this spatial transcriptomic data. We're also interested in design algorithm in this kind of single cell data. And another topic we are interested in is how to actually using cancer image data to design some deep learning method to do classification. Okay, thanks.
Thank you, Maisie. This is great. Thank you so much. Any questions from the audience? So Maisie, a quick question regarding the 10x genomics de novo assembly work. So compared to the, I think it's long ranger, right, or some older version of a pipeline. So the N50 or the facing blocks that you can achieve is compared to theirs, is better or?
It's better, and the switch error is much lower. Yes. Much lower.
Okay. Yeah. So could that algorithm be used like in a single cell setting? I think in the transcript level, if I don't know, but it's not a allele specific. Probably not.
You mean those apply to the single cell data? You mean?
Yeah, the single cell 10x genomics.
Yeah. Actually, that's actually a good question. I actually recently was reading some other a good paper from Nature method, which they using this. They use the phasing algorithm to better detect the CNV from single cell data, which I think is promising if we can involve some phasing information. Yeah. Another gossip question: why the tools starting with the Aquila? Yeah, so actually that's a very good question. I think why I feel everybody use kind of like relate to the star. I think sky this kind of like a tool. The lot was called like. So I was thinking, Aquila kind of like it's like my Yeah, it's always headache for us to name the tools. That's a good idea. Okay. Anyway, thank you so much. Thank you. So I would like to introduce our last speaker, Dr. Xu Wang from Auburn University. He will talk about single cell transcriptomes of analyzing circular tumor cells. So it's your time, Xu.
Thanks, Sir Chen. I'll jump right in. So the non-small cell lung cancer is the most abundant form of lung cancer. And at later stage, metastases are often occurring, and brain is a major target. Over 50% of metastases in CLC are in the brain. So the lung adenocarcinoma leptomeningeal metastasis is a common form affecting 5% of the lung cancer patients, and they often have poor disease outcomes. The CSF, or cerebral spinal fluid cytology, is the gold standard for diagnosis, and MRI was used to locate the solid tumor in the brain. So through lumbar puncture, we obtained 2 mils of CSF samples, and you can see that there's normal CSF cells, and there's also circulating tumor cells, or CTCs. We would like to know the transcriptome signature of CTCs in CSF samples to develop novel biomarkers or transcriptome markers for diagnosis. So what we did is from the CSF samples, we make single cell suspension, and we removed normal CSF cells using CD45 antibody. And the remaining CTCs were through FACS sorting, and we make one individual cell in each of the wells of 96 well plate, and went through SMART-seq2 sequencing protocol. For the sake of time, I won't go into the details of the experimental protocol. Before we do any analysis, we want to see whether we can capture the correct cell type composition in normal CSF samples. We enrolled 3 normal patients and get the CSF samples. They were tested because of different disease, which were known to not to be affected by the cell type composition. So we also include pure B cells and T cells. You can see that in normal CSF samples, there's no B cells. There's about 80% T cells and 20% normocytes with proper markers for different cell types. And this is the textbook percentage of T cells and monocytes in CSF samples, which serves as a proof of principle of our analysis pipeline. So we enrolled 5 lung cancer patients with signature of brain metastasis. And you can see here, the 3 normal control samples mainly have T cells and the monocytes, and you cannot find any tumor cells except for 14 cells of the P4 patients on their monocytes. So the tumor CTCs from individual patients form different clusters, suggesting heterogeneity among different patients. We need to be cautious because these samples are processed immediately after collection, and on different days, there could be technical batch variability. To exclude that, we take 2 different samples from the patient P1 2 months apart, and you can see that they form a coherent cluster, suggesting that the technical variability is not driving the heterogeneity we saw here in the t-SNE clustering plot. And for the patient CTCs, they are positive for lung markers, epithelial markers, and proliferation markers, which are signatures of tumor CTCs of epithelial origin. So you can see from this image that the CSF CTCs have a much larger diameter than the normal CSF cells. We were wondering whether we could handpick some CTCs from the CSF sample without any CD45 negative selections. And we did that not on purpose because we ran out of the antibody that day for patient P3. So interestingly, all the P3 cells we selected are monocytes. So suggesting that the CSF CTCs are very rare in our sample, and you have to have CD45 negative selection to be able to capture these CTCs. So the next analysis was the transcriptome signature of the CTCs compared to normal CSF cells. So these CSF cells from the patients, they have an immune score around 0, so suggesting they're definitely not immune cells. We also identified hundreds of upregulated genes as the signature of CSF CTCs. They were enriched for energy metabolism function, cell adhesion, and several tumor signaling pathways, which is not surprising. So we were also interested in the heterogeneity among different individual cells, and we plotted the correlation matrix among different cells based on a single cell transcriptome for our 5 patient samples and for a primary lung cancer cell line derived from human and 2 human-derived xenograft samples. You can see that for within patient heterogeneity, the CSF CTCs have a much higher heterogeneity compared to the primary tumor with lower cell-to-cell gene expression correlation. So when we were comparing inter-tumor versus intra-tumor heterogeneity, so the gene expression are highly correlated within each patient because they form clusters in the t-SNE plot and in the correlation matrix. These interpatient heterogeneity could be partially explained by spatial heterogeneity because the location for solid tumor is quite diverse from cerebral hemisphere or the temporal lobe or even in the cerebellum. So this spatial variability could contribute to the expression heterogeneity we observed. So we also investigated other characteristics of the CSF CTCs. The first is cancer stemness. We profiled the expression level of three different major stemness markers, CD44, ALDH, and CD133. You can see that expression level is quite variable. Not all patients, not all cells in all patients are expressing these markers, suggesting heritability. So the epithelial to mesenchymal transition is known to be the driver of epithelial tumor spreading. So we also checked the expression profile of these related markers. Almost all tumor cells have epithelial marker highly expressed here. And the extracellular matrix related markers are also abundant in all cells we checked. However, we observed the variable expression level of the EMT markers from no expression to highly expressed, suggesting a partial EMT. So this is also a signature of the CTC expression profile. The cancer testis antigens were also researched intensively because they were restricted in male germline in normal cells, but their expression were observed in many different tumor types. So we quantified the number of CTA genes in different patient samples. You can see we identified many significant differences. For example, the XAGE1B gene is mainly expressed in patient number 1, number 6, and number 7. BRDT1 is primarily expressed in P7. LY6K expression is observed in P4 and P6, and the SPAG9 is ubiquitously expressed in all patients we enrolled. So we also checked the cycling status of the CTCs comparing to primary tumor and a xenograft. What we found is that the CSF CTCs have a much lower proportion of high cycling cells, and the majority of the CSF CTCs are in the non-cycling state in the LUAD-LM patients. So we hope our transcriptome profiling could facilitate the development of novel markers for diagnosis of brain metastasis. So in the next few minutes, I would like to introduce you a special case of cancer of unknown primary. So this patient was included in the same batch. CUP cases are extremely difficult to identify the primary origin, but they're quite important because they account for 5% of the malignant epithelial tumors, and the patient outcome is extremely poor. So for this patient 8, we can locate the solid tumor in the brain, and physical examination, lab tests, and imaging procedures all fail to identify the primary location. So from our single-cell RNA-seq data in the clustering plot, P8 form a distinct cluster differentiated from other patients of lung origin. So the control samples are CD45 positive, and the other patient samples are all CD45 negative. So we want to know that is this tumor epithelial origin? Yes, it is, because the epithelial markers clearly demonstrate that P8, the P8 CTCs are epithelial origin, and so does the imaging work and staining work. So the proliferation marker CCND1 is also positive in almost all of the P8 cells. However, the P8 tumor cells were not lung origin. You can see that major lung markers are positive in all lung cancer brain metastasis patients, but not in patient P8. Now, you probably already noticed that we have 2 samples from P8, and they are clearly differentiated. These 2 samples were collected 6 months apart. So, and we observed some tumor progression in terms of the CTC transcriptome profile. And shortly after our second sampling, the patient passed away. So we want to know the origin of these CUP case, and we did staining for gastrointestinal cancer marker, prostate cancer marker, and liver cancer marker. They were all negative. And in order to elucidate the origin, we compared the gene expression profile of the lung cancer, lung origin CTCs and P8. And we identified a bunch of differentially expressed genes or significantly upregulated genes in P8. For example, PIP, ANKRD38, and SPINK8. So what are they? PIP and ANKRD38 are almost restricted in P8 with extremely high expression. They were known to be markers of prostate, breast, sweat, and salivary cancer. And we already know that it's not prostate cancer, it's not testis cancer. And the choices are breast cancer or salivary gland cancer. And we did a Cochrane gland staining and we get some positive results. So, however, we also checked histology from the salivary gland. So if it were salivary cancer, gland cancer, it's not the typical form of that cancer. So interestingly, we find high expression of this SCGB2A2 gene in 7 CTCs among hundreds of cells. And this is one of the power of single-cell RNA-seq. You will not be able to capture that in bulk RNA-seq. So this gene is a carcinoma marker for breast origin. So we suspect this cancer could have breast origin. So this was not tested because the patient is a male. So because the patient spouse refused autopsy, and we were never able to confirm the primary origin, but hopefully the single-cell RNA-seq could give us some hint about the origin of this CUP case. So this project is in collaboration with Dr. Zhang and Dr. Guan from Tongji University and Huashan Hospital in Shanghai. And the data analysis was done by a very talented graduate student in my lab. And I would like to thank all members of my lab and a few funding agents. Because we're in a vet school and this is what we really do. So now I'm ready for questions. Thank you.
Thank you, Xu. This is awesome. Fantastic work. So I will chime in for a quick question. You're working on the lung or some breast cancer, the metastasis, the brain metastasis, right? So yes. I will sequence all of the sites. This is actual the circular DNA, right? Circular RNA, and have you sequenced the primary site and the brain tumor and all of these sites, or just the circular RNA sites?
We tried. So we have the pharmacogenomics data of more than 500 loci, but we want to get RNA-seq from the primary tumor, which is a lung cancer. So bear in mind, they are stage 4 lung cancer patients. So we try to aspirate the cells from the primary site, but the library construction was not good. So the answer is we tried, but we don't have that sample right now.
Yeah, another question is in terms of origin of, I think RNA-seq sometimes maybe misleading. Have you checked I know single cell can do the single cell RNA-seq can do some copy number variations analysis. So from the copy number variation perspective, you may check the origin is from the DNA level kind of, right? That's maybe more straightforward for the evolution of these tumor cells. Have you checked the copy number variations and some other mutations?
We didn't, but that's a great suggestion. So also the DNA methylome could be very informative for the origin as well. So because we're working with human patients, so ideally we want to get samples from different body locations, but that's almost impossible. And we also want to move to some animal model to check this. But this is a great suggestion.
I think Nick Naven has some papers or some, and there's a method paper I thought is widely used. You probably can check out a little bit. So another naive question regarding the circulating cells, probably that's not easy to address. So the circulating tumor cells so the brain blood barrier and BBB, that's not an issue at all, right? It's a naive question. I don't know if the tumor cells within the brain and circulating out are different or
yeah.
Right?
So it's a great question whether the CTCs are different from the solid tumor in the brain. So that's a great question. So currently we are not 100% sure whether it's coming from the primary or lung origin. So it's definitely lung origin, but we don't know whether it's closer to the solid brain tumor or the primary lung tumor, and that's the next step. Yeah.
And also, I think I don't know if you have done that or probably compare with some brain metastatic data, like bulk data or some TCGA. TCGA probably doesn't contain these data sets, but some other data sets for like you identify some markers and if they are they can be cross-validated from other studies, something like that.
Yeah, absolutely. So we check with single RNA-seq data from in the public domain from primary human lung cancer. So, but the brain tumor single-cell RNA-seq data is very limited. Yeah, absolutely.
Okay. That's great. Thank you. And thank you for all the speakers' contribution. That's really awesome.